Hello, and sorry guys, I just rewatched John Campbell's video about mRNA nanostructures and I just can't stop laughing. Let's try again. Hello, and welcome to Back to the Science. I'm Dr. Susan Oliver, and I'm a scientist, and this is Cindy Oliver, and she's a dog. I have kind of given away what this video is about, so let's get straight into it. John Campbell recently published the following on YouTube and the platform formerly known as Twitter. Several friends were uncomfortable with this paper on nanostructures. I have therefore deleted my video report on this paper until its scientific validity or otherwise is clarified. It is good that we have such a global community of expertise to advise in near real time. Now, when John says his friends were uncomfortable, he means they were concerned that John's video was so stupid it would make all anti-vaxxers look like complete and utter idiots, as opposed to just looking like general misinformation spreaders. Now, we'll get on to the paper, which has to be one of the most hilariously bad papers that I have ever read. But believe it or not, John's video is even more stupid than the actual paper. Let's listen to the start.
A warm welcome to this talk. It's Friday the 6th of September. Now, I've heard about this phenomena years ago, of course, but only just got some evidence to support it, so we can only just report on it now. This is from peer-reviewed literature.
Now, Cindy did a bit of peer reviewing this morning, but Cindy's peer reviewing is a different kind of peer reviewing. To start with, it's a much higher quality of peer review than the peer review undertaken for this paper. According to the journal, normally at least 2 reviewers from the editorial board will be called upon to examine and critique materials in the subject matter areas of the journal. Technically complex and abstract theory and research may involve multiple back and forth interactions asking for detailed revisions, clarifications, and the like, with 3 to 7 reviewers examining and critiquing the work. Now, for those of you who don't know, peer review is not generally done by journal editors. The editors will give the paper a once-over to make sure it's not total bollocks and that it is of sufficient quality to be included in the journal, but it is then sent out to experts not associated with the journal to do the peer review. They don't do this at International Journal of Vaccine Theory, Practice and Research because it's not actually a real journal. It's an anti-vax mouthpiece, and the editors aren't qualified to even determine whether the papers they are receiving are bollocks or not. Further evidence of the fact that they are willing to publish any anti-vax bollocks is this editor's note that was actually added to the paper that we're talking about. The paper by Mead et al. Was retracted soon after the journal Curious published it. Part 1 of the 2-part response to that unwarranted retraction has already appeared in IJVTPR, and Part 2 will soon follow. Now, for anyone who doesn't know, the journal Curious is well known for publishing some pretty questionable papers. A paper has to be really bad to be retracted by them. Only an anti-vax mouthpiece would publish papers claiming the retraction wasn't warranted.
And it's scientists based in Japan and South Korea.
No, there weren't any scientists involved. There were a practicing physician with a speciality in obstetrics and gynecology and a professor of applied linguistics.
And what they've done is they've taken COVID vaccines, mostly Pfizer and Moderna, and cultured them, incubated them, to try and duplicate the conditions in the human body.
Let me just read you a bit of the methodology that they used. A minute sample, 0.03 millilitres of each injectable product, was added to normal saline, sterile distilled water and Hartmann's solution. each contained in a separate culture dish, either a 1.2 milliliter for embryo culture dish, 5-welled culture dish, or an 8 ml for Petri dish at room temperature, 15 to 25 degrees Celsius, adjusted for seasonal changes without any special added materials. How can John possibly think that bears any resemblance to the human body? To start with, the human body is generally just below 37 degrees Celsius, not between 15 and 25 degrees Celsius. And none of the liquids that the vaccine has been added to would be at physiological pH. Now, I'm sure someone is going to point out that they did actually do a short experiment where they placed some culture dishes on a hot plate set at 36.5 degrees Celsius, and therefore John isn't completely stupid. The correct way to keep a sample at biological temperature is to either place it in an incubator or an oven set at the correct temperature. You don't control the temperature of a sample by sitting it on a hot plate.
Now, I don't expect you're going to watch this video or be allowed to watch this video, but I'm going to do it anyway. And if by some chance a few of you actually get to see it, then that's brilliant. Now, here's the equipment that was actually used here. It's stereo microscopes. Now, basically all this means is you're looking at it with two eyes, therefore you get stereoscopic vision. And what happened was that initially they developed two-dimensional nanostructures, and then some became three-dimensional as well. And of course, you can see that with a stereoscopic microscope. Let's go and look at some now. That was the equipment that they were using. And of course, these days, you don't so much look down the microscope, it all goes on a screen, so you can take copies of it. Now, these are from the publication. These are some of the nanostructures that were observed.
Now, as some of you may know, I recorded a video with John in June 2021 before I even started this channel. I will just play you the end of that video.
I'm convinced, and Susan is a proper scientist. She is the real McCoy. So I'm accepting that explanation 100%. Susan, that's great. Thank you as always. Now, just a quick trailer. Me and Susan have got a slight deal going on here. We're going to talk about her scientific life in another video. So keep an eye out for that. We'll be talking about Susan's work in nanomedicine. And given that I haven't got a clue what nanomedicine is, I'm personally looking forward to that. So that'll be out sometime soon. So keep an eye out for that. Susan, wonderful to talk to you. Thank you for that update from Australia, and stay safe for the next 2 weeks.
Thank you, John, and you too.
Thank you.
Now, of course, that second interview never happened. John ghosted me after I sent him a polite email about a new ivermectin meta-analysis that clearly showed there was no evidence that ivermectin worked for COVID. Of course, I can only guess why John ghosted me, but it is worth mentioning that he had recently discovered that he could get a lot more views by talking up ivermectin. Anyway, if he had done the interview, he could have learnt a few things about nanomedicine. Like, for instance, the fact that you can't see nanostructures with an optical microscope because they are way too small. You need an electron microscope to see them. And these are just some pictures that I took with a transmission electron microscope of some nanoparticles that I made.
So these are the sort of structures that they were finding. I mean, what the heck is that? You know, that is a structure that spontaneously sort of put itself together, a spontaneous assembly of this structure from the COVID vaccine cultures.
You're not going to believe this, but I saw a similar picture to this in a public toilet today. It's almost like there's a grassroots effort to get the message out there because mainstream media, who are instructed by lizard overlords, are trying to suppress it. And a number of people have commented on John's post suggesting not dissimilar things. This tells me that John is being threatened. Stay strong, John. Don't let them intimidate you. Don't give in to pressure. I had heard that before and you just validated it. Yes, it's scary, But truth must be told. That's censorship, John. We need to look at it from all angles. Censorship stifles truth. You can see why John is so successful with his totally bollocks videos. He knows what his audience wants. Now, you may be wondering what the paper's authors were actually seeing under the microscopes. Before we talk about that, let's just laugh at John for a bit longer.
Let's look at a couple more pictures before we look at the text. So these spiral ones seem to come up, again, spontaneously, just put themselves together, spontaneous sort of, another spiral there, another one there. Tell you what, I don't like the idea of these spontaneously forming in the cells of my body, if that is indeed the case. We don't know that, but if that's the case, I don't like the idea of it at all. Not at all. And 10 micrometers, that's actually pretty big, actually. If that's the scale there, 10 micrometers. So this whole thing is actually quite large, relatively speaking. And what is that? Spontaneously formed structure. Well, that one.
Now, before people start sending me the names of papers about self-assembling nanoparticles and nanobots, let me just be clear, they are a thing. They just bear no resemblance whatsoever to the structures that John is showing pictures of, and they don't form in that way. Self-assembly is a pretty standard way of making nanoparticles. It relies on using some kind of macromolecule that has a hydrophilic, meaning water-loving, head, and a hydrophobic, meaning water-hating, tail. In its simplest form, it works in a similar way to the way that detergent encapsulates oil and appears to dissolve it. The self-assembly process is also used to encapsulate mRNA in lipid nanoparticles for the mRNA COVID vaccines. Although in this case, the lipid molecules form a double layer with the hydrophobic tails in the middle and the hydrophilic heads facing both the solution and the mRNA. You can also design nanoparticles to change behaviour based on pH, temperature, light, magnetic forces, etc., etc. It's all based on clever chemistry. There are no real robots involved, and by definition, they are nano-sized. So these pictures obviously aren't self-assembling nanostructures. So what are they? Well, it's impossible to say for sure without further analysis, but possibilities include dust, lint, dander, microplastics, salt crystals, bacteria, and fungi. And obviously, different pictures could be different things. And I haven't shown you all the pictures on this slide because they just had heaps and heaps of crazy pictures in the paper, so I've just sort of taken some of them. And there does appear to be some evidence for at least some of the samples containing either bacteria and or fungi, because the authors note that Hartmann's solution, calcium hypochlorite, hydrogen peroxide and chlorine dioxide were less accommodating according to their results in triggering and enabling the self-assembly of foreign materials than were normal saline and distilled water. All the solutions that were less likely to supposedly enable self-assembly just happened to have antimicrobial effects, which would have limited the growth of bacteria and fungi. Could, of course, be just an amazing coincidence, though. Now, the authors claim that there couldn't possibly be any bacterial or fungal contamination, and I'll just read out to you what they said about it because it's pretty bloody funny. Throughout the entire incubation period, which exceeded 1 year, no signs of bacterial or fungal contamination were observed in any of the mRNA vaccine specimens, Pfizer and Moderna. This is noteworthy because we did not rely on periodic exposure to any of the typical antibiotics or antifungal agents that are common to basic incubation studies. Because we were persuaded from direct microscopic observations in an earlier short-term incubation study, not presented here, that these foreign materials are not organic, but rather at least synthetic hybrid organisms or possibly animate robotic structures, we adopted an atypical approach to incubation. Our approach differs from usual culture methods, which periodically supply laboratory agents such as antibiotics and antifungal agents or other antibacterial or antifungal disinfectants that can dramatically alter the purity of the materials under observation. Growth in our studies was also maintained in small containers, but without any added O2 or CO2 supply. Okay, so they did nothing to control for microbial growth, but they're convinced it didn't happen because they didn't see any evidence. If you want to know if a solution is contaminated with bacteria or fungi, you don't just look at it. You need to properly test it. One way of doing this is by placing a drop of the solution on an agar plate and incubating it for about 24 hours. If the solution isn't contaminated with bacteria or fungi, the plate will stay clear, as you can see is the case in some of the quadrants on this agar in the picture. If it is contaminated, you will see growth on the plate, as you can see on some of the other quadrants. Unless you've done this or a similar test, you have no idea whether your solution is contaminated or not. All the solutions that were placed on the agar plates that are used in this picture looked clear to the naked eye, but as you can see, some were still contaminated. Now, the other question that you may have is how did these samples get contaminated in the first place? Again, there are lots of possibilities. Firstly, we know that the vials were primarily discarded vials that had already been used, so they easily could have been contaminated through use. We also know that the samples were added to plastic petri dishes, which of course would be a source of microplastic contamination as well as other types of contamination. Finally, the petri dishes were topped up with liquid weekly over a period of over a year. Every time this happened, there was opportunity for contaminants to be introduced, not to mention lots and lots of salt to form crystals in the saline samples. And of course, contamination could also have occurred during preparation of the slides for microscopy. Back when I used to work at a university, in addition to doing my own research, I used to sometimes run lab days for primary school students. The type of research that they did in this paper kind of reminds me of the research projects that the kids did in the lab. Actually, that's a bit unfair to the kids. Their research made much more sense. And I will just read you one more section from the paper because it really is just diabolical. Supplementary fluids and other known or potentially toxic materials that we studied with respect to the motility of sperm samples included soju, Korean distilled spirit 20 proof, red wine, beer, and silica water. Also, adding them to the normal saline solution as a control, we examined the impact of vitamin C, mica, and the incubation media of skin extracts taken from an injected recipient. We expected that the degree of cytotoxicity of any of the milieu examined would show up in the motility of the sperm cells from the various donors. I wonder. Anyway, I'll leave it there. The paper is bonkers, and John's video about it is even more bonkers. So bonkers that even his anti-vax mates had to pull him aside and have a quiet word. Now, I usually do my blurb now where I tell you if you'd like to look further into the data I presented, I provided links in the video's description. But in this case, I haven't really presented any data. I've just more talked about stuff that I know. So there won't be a lot of links. However, please remember this video is about the science, but you shouldn't take it as medical advice. For that, you should speak to your medical practitioner. If you've got this far, thank you for listening. And if you've liked, shared, or commented on the video, double thank you, because that helps the algorithm and means that more people will see the video. And we want as many people as possible to see this video since as John's deleted his video. Everyone needs to know how ridiculous he is. And of course, thank you to everyone who has bought me a coffee or little Cindy here. Say hello, Cindy. A treat. We really appreciate your support. We will be continuing to make videos about the science in the future. So if you'd like to join the cool kids and stay informed, please hit the subscribe button. Thank you.